الأصدقاء الأحباب، السلام عليكم. في هذه الحلقة الرابعة والستون سوف أتحدث بعمق أكثر ولفائدة طلاب وباحثي التغذية عن الجلايكوليسيس، وسوف تكون باللغة. أرجو المتابعة لمن يحبون المعرفة. أولا لأبدأ، there are some keywords that should be noticed in this session. First of all, let us know where this word glycolysis comes from. The word glycolysis is derived from the Greek word glykais, which means sweet, and the word lysis means to split, which means to the splitting of one glucose molecule into two molecules of pyruvate, which is the end product of the glycolysis. In the absence of oxygen, pyruvate usually enter the mitochondria, where it is oxidized to acetyl-CoA, whereas in the absence of oxygen, pyruvate is reduced into lactate. It's a simple definition of the word glycolysis. What about the keywords? Glycolysis, Krebs cycles, metabolism, and we find catabolism and anabolism. Anabolism is one of the metabolic pathways that construct mitochondria like DNA or RNA from smaller units. This reaction requires energy known as endergonic process, while anabolism is the building up aspects of the metabolism, whereas catabolism is the breaking down aspects. Anabolism is usually synonymous with biosynthesis. Glycolysis is the metabolic process that serves as foundation for both aerobic and anaerobic cellular respiration. In glycolysis, glucose is converted into pyruvate. Glucose is a six-member ring molecule found in the blood and is usually as a result of the breakdown of carbohydrates into sugars. It enters cells through specific transporters proteins that moves it from outside the cell into the cell's cytosol. All of the glycolytic enzymes are found in the cytosol. One point here I should talk about, that all enzymes are proteins, but not all the enzymes but not all the proteins. All the enzymes are proteins. This we should keep it in mind. In glycolysis, there are 10 steps. Okay, to continue, let us talk about the 10 steps in glycolysis. The first step is ruled by the enzyme hexokinase, where there is a conversion of D-glucose into glucose 6-phosphate. The enzyme that catalyzes this reaction is hexokinase. Now, we can see that the glucose ring is phosphorylated, and this phosphorylation is the process of adding phosphate group to a molecule derived from ATP. As a result, at this point in glycolysis, one molecule of ATP has been consumed. Now, atomic manganese is also involved to help shield the negative charges from the phosphate group. The result of this phosphorylation results in a molecule called glucose 6-phosphate. We indicate to it as G6P. The second step is the phosphoglucose isomerase. Here, what happened that there is a rearrangement of glucose 6-phosphate into fructose 6-phosphate, F6P, by glucose phosphate isomerase enzyme. Which is phosphoglucose isomerase. Here, the glucose 6-phosphate converted to fructose 6-phosphate, and this reaction occurs with the help of the enzyme phosphoglucose isomerase. It's solely an isomerization reaction. Step 3, the phosphofructose kinase. Here we can see the importance of the magnesium as a cofactor. Help in changes the fructose 6-phosphate into 1,6-biphosphate. This step is catalyzed by the phosphofructose kinase, converting the fructose 6-phosphate to fructose 1,6-biphosphate. Here once more I should make a notice on the activity of the manganese. The manganese, as I said before, is found in the chlorophyll of all the green vegetables and fruits. Step 4, aldolase, where we have 2 triphosphates. The enzyme aldolase splits the fructose 1,6-biphosphate into 2 sugars that are isomers to each other. These 2 sugars are dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. This step utilizes the enzyme aldolase, which catalyzes the cleavage of FBP, yield 2 carbon molecules, GAP and HDAP. Step 5, triphosphate isomerase enzyme. This enzyme converts the molecules DHAP and GAP. The glyceraldehyde is removed and used in the next step of glycolysis. Then GAP is the only molecule that continues in the glycolytic pathway. Step 6, glyceraldehyde 3-phosphate, GAPDH, dehydrogenase. In this step, the main events takes place that 1, glyceraldehyde 3-phosphate is oxidized by the coenzyme NAD, nicotinamide adenine dinucleotide. The second, the molecule is phosphorylated by the addition of a free phosphate group. The enzyme catalyzes this reaction is GAPDH. Step 7, phosphoglycerate kinase. This enzyme helps in transfers a phosphate group from 1,3-phosphoglycerate to ADP to form ATP and 3-phosphoglycerate. Step 8, phosphoglycerate mutase. In this step involves a simple rearrangement of the position of the phosphate group on the phosphoglycerate molecule. Now in step 9, enolases. These enzymes remove a molecule of water form from 2-phosphoglycerate to form phosphoenol pyruvic acid. While in step 10, pyruvate kinase, this enzyme transfers a phosphate from a phosphoenol pyruvate to ADP to form pyruvic acid and ATP. Here we can notice steps 1 and 3. We find 2 ATP had been involved in the constructions, while steps 7 and 10 we find generation of 4 ATP. Then the net visible result of ATP produced will be 2. To summarize all the steps, we find that enzymes got involved in glycolysis are, but here we should notice once more, all the enzymes are protein, but not all the proteins are enzymes. In step 1, we find hexokinase. In step 2, phosphoglucose isomerase. In step 3, phosphofructokinase. In step 4, aldolase. In step 5, triose phosphate isomerase. In step 6, glyceraldehyde 3-phosphate dehydrogenase. In step 7, phosphoglycositrase kinase. In step 8, phosphoglycositrate mutase. In step 9, enolase. While step 10, pyruvate kinase. In total, as a result, in the process of cellular respiration, a single molecule of glucose can yield up to 36 molecules of ATP. I hope with this simple illustration of glucose can help you to understand what is meant by glycolysis and the metabolism aspects, catabolism and anabolism. Thank you, and I hope you share this with all your colleagues. Thank you once more.